Fusion Mobile, quality e-learning experience on the go.
Some properties and functions of the cell. Movements that are due to external stimuli may be nastic, tactic, or tropic movements, and as such, there are types of responses. 1. Phototropism. 2. Hydrotropism. 3. Geotropism. 4. Thigmotropism. 5. Scototropism. 6. Chemotropism. For phototropism, this involves response of plants to light. The shoots are positively phototropic while the roots are negatively phototropic. Hydrotropism. This involves response to water. The roots of plants are positively hydrotropic while the shoots are negatively hydrotropic. Geotropism. This involves response of plants to gravity. The roots are positively geotropic while the shoots of plants are negatively geotropic. Thigmotropism. This involves response of the organs of certain plants to touch. Example: the stems of yam and morning glory twine round the support as a positive response to touch. Root tips grow away from stones as a negative response to touch. Scototropism. Response to shadow is referred to as scototropism. For example, vine seedlings grow towards the shadow of the nearest plant to it so as to climb round it for support. Chemotropism. Response of parts of a plant to concentrations of chemicals are referred to as chemotropism. There are differences between plant and animal reactions. In plant reactions, this involves movement of the parts of the body. Animal reactions may involve all parts of the body, as animals can move freely apart. Plant reactions involve shifts in growth rates or changes in internal water pressure. Animal reactions involve contraction and extension of muscles. Plant reactions have no specialized sense organs which receive stimuli. Animal reactions have special sense organs, as in higher animals which receive external stimuli. And lastly, plant reactions, effects of stimuli are transmitted by diffusion of chemical substances, and in animal reactions, effects of stimuli are transmitted by specialized nerve tissue. The following are movements. We have tactic movements, nastic movements, and also we have tropic movements. In tactic movements, this is the response of a whole organism towards or away from a stimulus. The stimulus, the name of response, and examples are stated in the table present. Light. The name of response is phototaxis. Examples are Euglena and Climadomonas swim toward light of low intensity, but away from light of high intensity. Chemical. The name of the response is chemotaxis. In most plants, sperm swim towards the chemical produced by the egg cell. Another stimulus is temperature. The name of its response is thermotaxis. Example: motile bacteria swim from cold regions to warm regions. Nastic movement. This is a response exhibited by a part of a plant to non-directional stimuli such as light intensity, temperature, or humidity. For example, closing of the morning glory when the light intensity is low. Folding of the leaves of Mimosa pudica when touched. Closing of the leaflets of the flamboyant sleeping movements due to low light intensity. For tropic movements, when a part of a plant moves in response to a directional stimulus, it shows a tropic movement or tropism. Tropisms are very slow growth movements. They are named according to the stimuli. Example: phototropism, response to light; hydrotropism, response to water. Movement. Cyclosis. This is continuous and uniform flow of cytoplasm seen in plants and amoeba. In animals, cyclosis occurs in protozoa like amoeba, amoeboid movement. Organs for movement. 1. Flagella. 2. The cilia. The flagella. They are long, few, and are used in moving in liquid medium by flashing against water current. Euglena and Chlamydomonas have flagella. The cilia. They are usually numerous and short. They aid movement in water, just like flagella. Growth movement is regulated by auxins and other plant hormones. Different plant hormones have different functions, as can be seen in the table. The plant hormones and its functions. We have auxin, 2. Gibberellin, 3. Cytokinin, 4. Florigens, 5. Abscisic acid. Its functions are stated in the table. You can as well pause this video to take note of its functions. Reproduction. This is the ability of living organisms to produce new individuals of their type. There are 2 types of reproduction. 1. Asexual reproduction and 2. Sexual reproduction. For asexual reproduction, we have the following: 1. Binary fission, 2. Budding, 3. Spore formation, 4. Fragmentation, 5. Vegetative propagation, that is artificial and natural. And for sexual reproduction, we have 1. Conjugation, 2. Fission of gametes. Asexual reproduction involves a single individual which produces an offspring by itself without the fission of nuclei. The individual cells involved divide by means of mitosis. Offspring which are identical, that is clones, to the parent cells are always produced. Asexual organism is common among simple organisms and flowering plants. Sexual reproduction. Sexual reproduction is a type of reproduction which involves the fission of the male gametes and the female gametes to form a zygote, which later develops to a young offspring. So basically, the difference between an asexual reproduction and a sexual reproduction is, for asexual reproduction, it involves just one parent or single individual, and for sexual reproduction it involves the fission of male and female gametes which form a zygote. The types of asexual reproduction include the binary fission. This is the simplest form of asexual reproduction, during which a parent organism divides into 2 parts, each of which exists by itself. Example: bacteria and protozoa such as amoeba. In some other type of fission, a cell may divide into more than 2 parts. The budding. The new offspring formed develops as a protrusion or outgrowth of the parent. This outgrowth is referred to as a bud. The bud may form on an internal or external buds, break up from the parent without inflicting an injury to lead an independent life. Examples of these are in yeast, hydra, coral, and polyps. Spore formation. Certain plants such as fungi produce spores, which are small unicellular bodies. They are light and can easily be dispersed by air. Each spore can develop into an independent organism. This type of formation may be called sporulation. Another is fragmentation. A part of an organism may break up or fragment and give rise to a new organism. Example of this may be found in Spirogyra, sponges, and coelenterate. Vegetative propagation. This is of two types. We have the natural and the artificial. This occurs in higher plants, during which a new plant grows from any portion of an older one than the seeds. Stems and roots are modified for vegetative propagation. Artificial vegetative propagation. This is carried out by planting parts of perennating organs, that is, organs that survive from one growing season to the next, like tubers and rhizomes. Each perennating or must possess a bud which can develop into a new plant. Method of artificial vegetative propagation. The methods include the following: 1. Layering. 2. Cutting. 3. Marcotting. 4. Grafting. Layering. A healthy branch of a plant is bent until it touches the ground. The free end is tied to a strong support and fixed to the ground. The bent portion is fixed in place by 2 pegs and it is covered with soil. After some time, roots are given off from the cut from the parent plant and allowed to grow on its own. It is used in plants such as cocoa, rose, and bougainvillea. The second method of artificial vegetative propagation is cutting. Healthy portions of a branch of a stem is cut. The cut end is treated with rooting hormone to promote root growth. The cutting is then planted in a shady space. Examples are cassava, croton, and sweet potato. Marcotting. With a knife, a ring is made round a portion of the bark of a healthy branch of a plant. The bark within the ring is removed. Note that the bark consists of the phloem tissues that translocate food materials. Soil rich in humus is molded round the ring and it is held in place by the coconut husks. After some time, roots are given off from the ring area and the branch is then cut off. The husks are removed and the rooted branch is then planted. Examples are mango and lemon. For grafting, grafting is closely related to budding and it is a method of artificial vegetative propagation which involves the application of a whole school or stem into another plant. Grafting brings into close contact the vascular cambia of both the scion and the stalk. Plants easily propagated by grafting is mainly the citrus, example, orange, grape, tangerine, etc. Sexual reproduction. 1. Conjugation. 2. Fusion of gametes. Conjugation. Conjugation is a simple type of sexual reproduction which occurs in some lower organisms such as mucor, Rhizopus, Paramecium, and Spirogyra. Conjugation is a process by which nuclear material is passed from one cell to another. In Spirogyra, for example, conjugation starts with 2 filaments called conjugants coming together to lie side by side. Lateral outgrowth starts to develop opposite each other from opposite cells in the 2 filaments. The outgrowths meet and the separating walls dissolve to form a continuous channel called conjugate tube. Meanwhile, the cytoplasm of each cell shrinks away from the cell wall and round up to form a gamete. Fusion of gametes, as well called fertilization. Fission of gametes is the union of the haploid male and female gametes to produce diploid organisms called zygote. The process of this union of the gametes to form the zygote is called fertilization. In higher animals, These gametes are formed in special organs called gonads. The male gametes, called spermatozoa or simply sperm, are produced in the testes by an erotic cell division called spermatogenesis. In like manner, the gametes called ova or eggs produced in the ovary also by an erotic cell division called oogenesis. Fission of gametes, called fertilization, is the union of the haploid male and female gametes to produce diploid organisms called zygotes. This is evident in the diagram. Comparison of vegetative and sexual reproduction by seed. This comparison include, for vegetative propagation, there are uniform offsprings, and sexually by seed, offsprings are not uniform. For vegetative propagation, only a single plant is present, but sexually by seed, 2 parents are required. In vegetative propagation, pollinating agents required, and sexually by seed, pollinating agents are required. For vegetative propagation, the plants spread faster, and sexually by seed, plants spread slowly. For vegetative propagation, offspring survive adverse conditions easily, and sexually by seed, germinating seeds often die under adverse conditions. Vegetative propagation, a limited dispersal is witnessed, and for sexually by seed, dispersal is unlimited. For vegetative propagation, diseases are transmitted from one generation to another, and for sexually by seed, transmission of diseases to the next generation is very rare. For vegetative propagation, uniform nature of offspring does not give room for evolutionary advancement, and for sexually by seed, varied nature of offspring gives room for evolutionary advancement. Meiosis. Meiosis is the form of division which brings about a halving of the chromosome number as well as an interchange of genetic materials between homologous chromosomes. In animals, meiosis takes place at the formation of the gametes, and in diploid plants at the formation of spores. It consists of 2 divisions. First, meiotic division. A. Pairs of homologous chromosomes lie close together, each already composing of a pair of chromatids. B. Separation takes place except at chiasmata, with chromatids exchanging material by breaking and reforming at the chiasmata, that is, crossing over. C. Nuclear membrane disappears and spindle forms. And D. Chromosomes migrate to ends of spindle, taking exchange materials with them. The second meiotic division, the following occurs. A. The chromosomes divide again at right angles to the first division. This causes the chromatids to separate. B. 4 new daughter cells are formed, each containing the haploid number of chromosomes. The differences between meiosis and mitosis. For meiosis, haploid number of chromosomes result after division. For mitosis, diploid number of chromosomes are formed after division. For meiosis, 2 stages of division, and mitosis 1 stage of division. 3. 4 daughter cells are formed, and for mitosis 2 daughter cells are formed. 4. For meiosis, it occurs only in sex cells, and for mitosis occurs in body or somatic cells. 5. Exchange of genetic materials is due to crossing over in meiosis, whereas in mitosis, no crossing over, no exchange of materials. 6. For meiosis, 2 whole chromosomes form bivalent, and for mitosis, no formation of bivalent. 7. Chromosomes are arranged in pairs in the parent cell, but new cells have only one of each homologous pair. and for mitosis, chromosomes are arranged in pairs in both parent and new cells. 8. There is exchange of materials that result in variation in meiosis, but for mitosis, there is no exchange of material between the chromatids.